So we're going to be going over Unit 2, Cells and Microscopes. One thing to remember during this is I'm going to be talking and I don't know if you're going to be done with your notes. So you can pause this as you need to get down the notes and then listen to what I say. So don't forget to pause if you need to. So, cells under the microscope. First, two big scientists that had anything to do with the microscope were Robert Hooke and Antoine van Leeuwenhoek. Robert Hooke, he observed a thin slice of cork underneath a microscope in 1665. But before then, no one really used microscopes. They weren't established, they weren't around, so it was really hard to see the world microscopically as we know today. At that point, it was still kind of a blurry image. He noticed a lot of little bitty boxes within that cork, and he decided to call them cells. So that's how we get the term cells. Antoine van Leeuwenhoek, he viewed water from a pond under a microscope, and he noticed tiny living creatures and called them animalcules, or tiny animals. So both of these scientists had something huge to do with microscopes. Here's a picture of both of them. Here is a picture of Robert Hooke. This is the slice of cork, very, very, very thin slice of cork, and you can see the little boxes in here. And then the little boxes are the cells that he coined the term for. This is a picture of the microscope that he used. So the specimen was down here, and he looked through this and had to reflect light off. Now we just flip a switch and we have light. So it was 1665, very, very basic. Here's Antoine van Leeuwenhoek just posing for his picture. So, characteristics of microscopes. We have a light microscope and an electron microscope. We do not have an electron microscope here at school. We will only be using a light microscope in this class. A light microscope is a microscope that uses a beam of visible light passing through one or more lenses to magnify an object. So one or more lenses. The microscopes that we use, there's a lens that you put your eye up to, and then there's a lens that you can switch and change the power. So that's our two lenses that we use here at school. The electron microscope, same thing, but it uses electrons to magnify objects instead of lenses and light. Magnification and resolution. Many people get these confused. You need to make sure that you keep them straight. Magnification is increasing the object's size by using lenses or mirrors. Resolution, however, is making these details very clear. Early on in history with microscopes, they could magnify things, but they were really, really blurry, so they didn't have resolution. Nowadays, we can magnify them to make them large and then focus in on the very fine details because we have the ability to adjust our resolution. Very key point to have both to see details. So, right now, discuss amongst yourself the difference between magnification and resolution. See if you can put it into your own words. Go ahead and pause now and discuss. Cell theory. The cell theory is a theory that states that all living things are made up of cells, that cells are the basic units of organisms, and that each cell in a multicellular organism has a specific job, and that cells come only from existing cells. So there are three key points to the cell theory. Each one of these dots is a key point. The first one being all living things are made up of cells. Okay, that's point number one. Point number two, all cells are the basic units of organisms, with each cell having a specific job. And point number three is that that cells come only from existing cells. They don't just poof appear out of somewhere. Small cells function more efficiently than large cells. You have seen this exact slide last unit. This is how important the cell size is, that it's in 2 different units for you, okay? Don't forget we have about 100 trillion cells in the human body. They are very small, 5 micrometers to 20 micrometers in diameter. The reason small cells function more efficiently than large cells is because there's a larger surface area to volume ratio. You've seen this slide too. When I say larger surface area to volume ratio, I'm talking about this number right here. There's a bigger difference between 6 and 1 than there is 3 and 2. And this, I feel like, is the best picture we've already talked about. This is all 64 millimeters cube volume, each one of these. But because this has a smaller surface area to volume ratio, because these numbers are close together, so it's a smaller surface area to volume ratio, the information cannot be exchanged as quickly. But when you come over here, where the surface area to volume ratio is larger, all of this information can be switched back and forth in all these little nooks and crannies quickly so cells can function as efficiently as possible. And we went over this example. Which one's going to melt quicker? The bag of ice cubes or the block? We've gone over this. Just a little reminder. Okay. Cell types. There are two cell types that we're going to be talking about. We've got prokaryotes and eukaryotes. Prokaryotic cells, they have no nucleus, no membrane-bound organelles. They are really small, 1 to 10 micrometers in size. They evolved 3.5 billion years ago, and these are bacteria. So if I say, what kind of cells are prokaryotic cells? Your answer is bacteria. These are very, very, very simple, very small cells. Then you have eukaryotes or eukaryotic cells. They do have a nucleus. They do have organelles. They are larger. They're still small, but they're larger, 2 to 1,000 micrometers in size. Evolved 1.5 billion years ago, and this is everything other than bacteria. So let's talk about prokaryotic cells first. A prokaryotic cell is an organism that consists of a single cell. It's a single cell. That's important to know that prokaryotic cells are single cells and they are bacteria. That is hugely important. I pinky promise you it will be on the test. An organism that is consistent of a single cell that does not have a nucleus or cell organelles. An example of a prokaryotic cell would be a bacteria. So they're very simple. They have no nucleus, no organelles, and they are a bacteria. This is them. They have got different cilia and flagella on the outside to help them with movement, but other than that, their genetic material is this DNA, clumped up in a big wad in the center. They do have ribosomes, and that's it. They are very simple, very small, and they are bacteria. So prokaryotic cells, very small, very simple, no nucleus, no membrane-bound organelles, very simple. So, characteristics of these prokaryotic cells, they cannot carry out specialized functions. Since they are so simple, they cannot be specialized. Prokaryotic cells can exist in a variety of environmental conditions. Genetic information is normally in a circle that contains DNA, and it is not located in a nucleus. Prokaryotic cells do not have a nucleus. I think I've said they don't have a nucleus now about 5 times. Pretty important to remember. Let's go back a slide. Look at this. Genetic information is normally in a circle that contains DNA. If you look at this picture, it's one big circle that's been blobbed together. There's nothing surrounding it, and that's where you find its genetic information or its DNA. Prokaryotes have cell walls, however. Cell walls, they are a rigid structure that surround the cell membrane and provide support to the cell. So first, you are going to have the cell membrane, and then you have this cell wall that goes around that. Some are then surrounded by capsules. So if you have a capsule, it's even a capsule around that. And this allows it to stick. Okay, so right here, this would be the cell membrane. This would be the cell wall. And this would be the capsule. So these prokaryotic cells are protected extremely by 3 different layers. Sometimes they also have flagella. Flagella are long hair-like structures that grow out of the cell and enable the cell to move around, just like a little tail. Eukaryotes. This is an organism made up of cells that have a nucleus enclosed by a membrane. They have multiple chromosomes and membrane-bound organelles. This is where we get complex. This is everything but bacteria. These are the highly specialized cells. So on to eukaryotic cells. And don't forget to pause at this point. Don't forget to pause. If you need to write these notes down, pause, because I'm just going through them, okay? I know you will not be able to keep up as fast as I'm going, so remember to pause. So this is what a eukaryotic cell looks like. Much, much, much different, much more going on than a prokaryotic cell. Much more. This, I'm sure, is a picture that you're used to seeing of a cell. This is also a eukaryotic cell. This first one is an animal eukaryotic cell. This is a plant eukaryotic cell. So eukaryotic cells can be then divided up into 2 more subclasses: animal cells and plant cells. So let's just talk about general eukaryotic cells first. The nucleus. It has a nucleus. It is a membrane-bound organelle that contains cells' DNA. And it has the role in the processes such as growth, metabolism, and reproduction. I've been saying membrane-bound organelle a ton. An organelle is something that is very small, similar to an organ in our body, but it's in a cell. My stomach has a specific function. My heart has a specific function. Those are 2 of my organs. Organelles are really tiny in a cell and have specific functions. Membrane bound means that it's not just floating around, there's something containing it. So a nucleus, all eukaryotic cells have, and it is a membrane bound organelle. Some eukaryotic cells have cilia, and these are hair-like structures arranged tightly packed together and project from the surface of the cell. They normally sweep mucus and debris out of the respiratory tract. So if you know someone who smokes, sometimes they have that smoker's cough. It's because that cilia sometimes lays down and doesn't act as well. So they've really got to get that coughing going because the mucus doesn't sweep out as well of their respiratory tract. Here we go. There's my definition of organelle. I was ahead of myself one slide. Organelles is a small body that are found in the cytoplasm of the cell and are specialized to perform specific functions. Organelles allow eukaryotic cells to perform more complex functions than prokaryotic cells. The cell membrane. So the cell membrane is a phospholipid bilayer. This is extremely important to know. A phospholipid bilayer. Know this, know this, know this. That is what a cell membrane is made up of, and it covers the cell's surface and acts as a barrier between the inside of a cell and the cell's environment. Cytoplasm is a region of the cell within the membrane that includes fluid and the cytoskeleton and all of the organelles. Cytoskeleton. Much like what you think it is, our skeleton provides us support and shape. The cytoskeleton provides the cell with support and shape. The cytoplasmic network of protein filaments play an essential role in cell movement, shape, and division. It provides an interior framework for an animal cell. It links one region to another and give support and structure. Much just like our skeleton in our body, the cytoskeleton of a cell gives structure and support. Ribosomes. Ribosomes are found in both prokaryotic and eukaryotic cells. This is a cell organelle composed of RNA and protein, and it is the site of protein synthesis. So the big function of a ribosome is the protein synthesis. That's the big function of a ribosome. Okay, more about the cell membrane. The cytoplasm in all the cells are contained by the cell membrane. So that's like the wall or the boundary of the cell. Cell membranes are fluid, not rigid like an eggshell, so they can move and squeeze in and out of places. They're not tough. Cell membranes, or the cell membrane is made up of lipids, specifically phospholipids. The way the phospholipids interact with the water determines how things can cross the cell membrane. You don't want just anything crossing your cell membrane. Just like we don't want anything, coming into our classroom, so we've got a wall around it or whatever. There's got to be a barrier. So phospholipids, this is a lipid that contains phosphorus and is a structural component in the cell membrane. It is arranged in a bilayer. Bilayer. So we've got this prefix right here, bi, bi. What do you think that means? Bi, the prefix bi, as you can see my lovely writing here, means two. So arranged as a lipid bilayer, the basic structure of a biological membrane, and it's composed of, ooh, look, there it is, 2 layers. So here is what one whole phospholipid looks like. It is made of 1 polar head and 2 nonpolar tails. Important to know. 1 polar head, 2 nonpolar tails. When they're arranged in a bilayer, it's 2 layers of them. So the tails hang out here in the center, and the heads are on the outside. And this is what it looks like in a larger version of them put together. So this is what the whole cell membrane is this phospholipid bilayer. And here's a bigger picture of it. You can see the nonpolar tails and the polar heads on either side. This is the inside of the cell. This is the outside of the cell. And the whole cell membrane has got these phospholipids all over it, as well as some other things running through it. But it's got all of these phospholipids bilayers all the way through it. Okay? And it moves. It's kind of like a wave. It moves as it needs to. So the cell membrane is selectively permeable due to the phospholipid bilayer. What do you think selectively permeable means? Selectively permeable. First thing is, what does permeable mean? Permeable means that something can go through the membrane. Now, what does selectively mean? Selectively means that the membrane chooses. So the membrane chooses what can go through. Selectively permeable. Various proteins are located in the membrane of the cell. The fluidity of the membrane allows for these proteins to move around the membrane. So let me go back a couple pictures. Hold on. These are these proteins that we're talking about. And because the phospholipid membrane is fluid, these guys just float around like they're on an inner tube in water and go wherever they need to go. Okay. those protein or cell membrane proteins are cell surface markers, and they say, hey, 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 I'm an arm cell. I'm not an eye cell. So it marks what the cell is. Receptor proteins. Receptor proteins, they can receive information and send that information on through. Enzymes can speed up the process. You'll see that. Transport proteins, they actually allow things to go through them that couldn't go through the membrane. Here they are. Okay, so we've got this cell surface marker. Okay, it says, hey, 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 I'm an arm cell, I'm not an egg cell, or eye cell, sorry. I'm an arm cell, not an eye cell, so it identifies the cell. Number 2 was the receptor protein. It's going to accept a message right out here, and the message will attach. And then it's kind of like a telephone call. Then the message is relayed into the inside of the cell, so the inside can do something. Enzymes, yet again, it speeds up those chemical reactions. And then you've got transport proteins. Something that couldn't just slice through the membrane will go through the transport protein instead. We're going to now talk about every specific organelle inside the eukaryotic cell. The first one is the nucleus. Right here, this is the nucleus, boxed in. So I'm going to try to box each one in so you can see it, and then I will have a slide over it. The nucleus, still pictured up here, functions in a eukaryotic cell. Most functions are controlled by the nucleus in a eukaryotic cell. This is the center point. This is where all the information is. It is surrounded by a nuclear envelope. So if you see this area right here, that's what it's surrounded by, and everything else in here is the nucleus contents. Channels called nuclear pores, that's these holes that my mouse is pointing to, are all over the nuclear membrane, so things can come in and out. This is where the DNA is stored, and DNA codes for hereditary information. Excuse me. The next one is the endoplasmic reticulum. The endoplasmic reticulum, and we're going to talk about ribosomes too, are found in eukaryotic cells. Cells make proteins on the ribosome. Cells make proteins on the ribosome. So ribosomes are the location for protein synthesis. Ribosomes are suspended in the cytoplasm of prokaryotic cells. But on eukaryotic cells, some are suspended in the cytoplasm, but others are attached to the endoplasmic reticulum. The endoplasmic reticulum is a system of membranes that is found in a cell's cytoplasm that assists in the production, processing, and transport of proteins and the production of lipids. So once these proteins are made, see these little bitty dots, kind of blurry, these little bitty dots are ribosomes, and it's going to make the proteins. Once they make the proteins, it goes into the endoplasmic reticulum. From there, inside the endoplasmic reticulum, these small vesicles are formed. And vesicles is a small cavity or sac that contains materials in a eukaryotic cell. So these proteins are going to be contained by a vesicle, a little package. It's going to be packaged up, and then the protein can go where it needs to go. The next organelle that we're going to talk about is the Golgi apparatus. The Golgi apparatus will package and distribute proteins, just like, very similar to the endoplasmic reticulum of the packaging, but this is now going to distribute. So newly made proteins are transferred to the Golgi apparatus, this little guy up here. The Golgi apparatus is an organelle that helps package materials to be transported out of the cell. These purple little blobs are the packages that have come off that are going to head towards the cell membrane to be released. A lysosome is another cell organelle, and it contains digestive enzymes. So it breaks down the trash and unwanted materials. And here's the lysosome right here. I don't know why I didn't have its own picture. But there's the lysosome. Little guy cleans up the cell. On to the mitochondria. The mitochondria is a cell organelle that is surrounded by two membranes. That is very important to remember, that there are two membranes of the mitochondria. And this is the site of cellular respiration, which produces ATP or energy. Cells that need more energy will have more mitochondria. What cells in your body would need more energy? Would a skin cell, if I can write skin, I thought I could quickly, but apparently not. It's going to look like a 2-year-old. Skin or muscle cell? A skin or a muscle cell? Which one would need more mitochondria? Which one needs more energy, skin or muscle? Muscle. Therefore, they need more mitochondria. Mitochondria also hugely, hugely, hugely important, contains its own DNA. So the two things to remember here is that mitochondria have two membranes and that they have their own DNA. That is huge. Ignore my skin comment. So those are the organelles specific to the animal cell. These three that we're about to talk about are specific to the plant cell only. In plant cells, you now have the cell wall. So the cell membrane of the plant is still there, but now a cell wall is around it. And this helps to support and maintain the cell shape. Here's the thing. In animal cells, we've got skeletons in animals to keep us formed in our shape. But a tree does not have a skeleton. So it needs this cell wall so it can stand up straight and tall and be big and bushy and provide tons of shade. But without that cell wall, the tree would just fall down. So it's kind of the plant's skeleton, for lack of better terms, in order for plants to stand up straight. Chloroplasts are little green organelles. They are found in plant and algae cells where photosynthesis occurs. We'll talk about photosynthesis more next unit. Chloroplasts, interestingly enough, have some stuff in common with the mitochondria. It has two membranes, and it has their own DNA. Don't you love my circles? If I had a mouse and not a trackpad, it would look better. So chloroplasts have two membranes, and they have their own DNA. Central vacuole. This is a large cavity or sac that is found in plant cells or protozoans that contain air or partially digested food. This makes the cell rigid and allows for plants to stand straight up. This also allows for trees and everything else to stand straight up with the help of the cell wall. Have you ever gone to the refrigerator and found some lettuce that's a little not as crisp as you would like it? You run it under cold water and all of a sudden it becomes crisp again. Has nothing to do, it has some stuff to do with the cold water, but what that is the water filled back up that central vacuole and it made it more rigid, so then you get another crisp lettuce instead of just some lettuce that was slightly wilted. That's a good reason. One, we want to wash produce off, but another reason is to fill up the central vacuole so we have crisp, nice vegetables when we go to eat. So, at this point, if I said, what's number 1? What's number 2? What's number 3? And that's actually what your assignment is going to be right now. What are all of these things? What is number 1 through 12? What is number 1 through 12? On a piece of paper now, please, I forgot 13. Number 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, and label what these organelles are by this picture. I want you to be able to identify them. Okay, 2 things. Number 3 is pointing at this little dot. Number 5 is pointing at this whole structure. Okay? So pause, do this, and turn this in. Now I'm sure that you have some questions. However, you can re-listen to this. You can pause this as many times as you need to.